So today's our first day on DNA. And so the first chapter is about structure and function. Then we'll talk about what information and how it's totally changed science. Anyway, so it wasn't until 1945 that they actually believed that DNA was a material coding for cellular identity. And there's some interesting stories I've heard from older scientists from Europe about how there was certain people who controlled countries who didn't believe in DNA. And yeah, it's kind of interesting. There's been some interesting, almost hilarious things done because of that. Anyway, so DNA stands for deoxyribonucleic acid. So it's deoxyribose. You know the structure of ribose. And it's an acid because of the phosphoric acid. And so anyway, we'll look at it in higher organisms first. So let me show you figure 1. So here's the DNA double helix. So you can see that it's about 2 nanometers in diameter, the basic thread. And then it's twisted around into what's called a nucleosome. So here's histone protein in the middle, and it's kind of wrapped around it. So when you take one nucleosome and that's this, you have a whole bunch of those, right? So the DNA is wound up around these nucleoproteins. And then that's further classified as a solenoid. When you have a whole bunch of them together like this, they start to create another alternating structure. But all this really has to do with protecting the DNA, because in higher organisms, it's extremely long, extremely susceptible to being broken and attacked by all kinds of other chemical and physical things. So then the solenoids are kind of wrapped into these loops. Okay, and the matrix, of course, is more protein. And then this gets turned around, so it gets this, what's called a mini band over here. So this is protein over here, and each of those little lines right there is a nucleosome. Then finally, if you were to have this and flattened out on this particular orientation, it would simply fold itself up until you get what's called a chromosome. So the chromosome has a tremendous amount of protein. And now let's say, for example, you're a molecule of RNA polymerase. Your job is to make messenger RNA from a gene. So you come up to this chromosome, it's just like, where the heck is the gene? That gene is so wound up. It's so tight. The data in here is so protected. It's like, how does that even happen? And of course, there are parts of the chromosome that are more susceptible to breakage, right? And so if you look at a chromosome, say a gene that's way buried in here, it's probably harder to get to than a gene that's out here on the surface. And then, let me just go back to this again. So then figure 2 shows the monomers that make up DNA polymer are high energy phosphoanhydrides. So this is one thing that most pictures of DNA just fail to point out. So here's 2 monomers, deoxy-ATP. And what is the difference between deoxy-ATP and ATP? Simply at the 2 position right here, there's no hydroxyl group. So this is 2-deoxyribose. You can see, oh, phosphoester, phosphoanhydride, phosphoanhydride. This is a base that's known as adenine, but it's a purine base. Here's a pyrimidine base. And so when these are put together by an enzyme called DNA polymerase, it directs how the bonds are formed. So there's a dehydration. So notice that this right here, this triphosphate right here stays intact, but this hydroxy group right here ends up replacing this pyrophosphate right here, this diphosphate, because it changes from this alcohol to this alcohol. So when you do that, this pyrophosphate or diphosphate goes off, and it later is hydrolyzed. So thinking about Le Chatelier's principle, if the immediate product in this equilibrium is diphosphate and you break it apart, it's no longer going to let the enzyme go in reverse direction. And then what kind of a bond do we end up with? Here's a phosphoester. Here's another phosphoester. This one was already in existence. But right here is where this attacks here. Wait, I'm sorry. Right here is where this attacks here. So we go from the 3' of one to the 5' of the other. Does that make sense? So what kind of a bond do we have? It's called the 3' to 5' phosphodiester bond. And what are we looking at? The convention is if we write a single length of DNA, the 5' M is always on the left. Okay, what's the 5' end? Well, here's the 5' of the ribose, right? So 5, 4, 3, 2, 1. So this is the 5'. It happens to be an A. And on the 3' over here, which you'll see, it would attach to another one over here. It's not shown, but in this case, it would be a C. So this bond right here is the bond right there. And as you probably know, because you're not new to DNA, the sequence is complementary. So in the second strand, if this is a T, that's going to be an A, always. If that's a C, that's going to be a G, always. So if I know the sequence of this, I can deduce the sequence of that. And you can see that they're parallel, but they're anti-parallel. I mean, it's not head to head, it's head to toe. It's an anti-parallel relationship. So I'm just going to show you the pictures first. So here we see the double helix, right? Every time there's a C in one strand, the G is opposite, T in the A, right? And so you can see that there's these grooves over here. The grooves are accentuated if you look at the space filling model. The minor groove is smaller than the major groove. And here's another picture of it. So this is showing you between a G and a C, and those little lines right there, those black lines are showing you are hydrogen bonds. So you have 3 hydrogen bonds possible between a G and a C, and you have 2 hydrogen bonds possible between a T and an A. Okay. So the other thing that you might be able to see from this is that those hydrogen bonds between the 2 strands are pretty much perpendicular to the length of the strand itself. So why do these 2 strands get pulled into this double helical shape? It's because that's what maximizes the hydrogen bonding between the 2 strands. Now, why isn't this moving? Here we go. It's moving. So here is another way to look at that. Here's an A and a T, right? So notice that there's two hydrogen bonds over here, shown in those little blue hash marks. And here's the actual distance between bases. So the distance of 11.1 angstroms is from here to here, right? Then between a G and a C, there's three hydrogen bonds. Here's the average distance between the two strands over here. Notice that those are fairly close. Notice also that you have a big ring across from a small ring in both cases. What that means, if you had two big rings across from each other, they'd be crowding. If you had two small rings across from each other, they wouldn't reach. So to get maximum hydrogen bonding is really between an A and a T and a G and a C. And so this is really what determines all the different properties of DNA. Hydrogen bonding between the strands. That's why if we know the sequence of one, we know the sequence of the other strand. Okay, well, of course you're going to stop me if you have questions, right? Come on. So let's see, what can we say more about that? So here's some characteristics. The two strands are anti-parallel, which is head to toe. The two strands are complementary. And the number of A's should equal the number of T's. And the number of G's should equal the number of C's. But all the other ones are not necessarily be equal. The number of purines equals the number of pyrimidines. So the purine is the big base, pyrimidines are the small ones. And so you have maximum hydrogen bonding between an A and a T and a G and a C. So the duplex twists into a right-handed helix, and that maximizes the hydrogen bonds. And then another stabilizing factor is that the hydrophobic bases are inside the helix away from water, or histone. And I guess I could say something about some of the terminologies that we use. When we say naked DNA, that's when the histone protein has been removed. Okay? So if you take naked DNA and put it in water, it dissolves in water. But the DNA itself in biology is really dissolved in histone and then suspended in the cellular matrix. So then the hydrophilic sugar phosphate backbone is outside of the helix, dissolved in water. And then we get ring stacking between bases. I'll go back and explain that in a minute. And then, yes, DNA is groovy. So let me go back and explain ring stacking a little bit. Where's the best way to look at it? Maybe here, okay? You see this base right here and that base right there? They are both aromatic. All the bases in DNA are aromatic. And that means that they have delocalized electrons. So if I have a delocalized electrons and some energy comes along and it causes these electrons to travel in, say, the counterclockwise direction, that creates a north and a south. So that same thing would happen with the base above it. There'd be a north and a south. So the south would be attracted to the north, then the south would be attracted to the next north. And that's a form of London's attractions. The wrong button there. Okay, so questions so far about the structure? So the central dogma of molecular biology, and yes, I was uncomfortable with that word when I first learned it as well. It's like, don't step in the dogma kind of a thing. Was really that DNA was turned into messenger RNA and then into protein. So I've embellished this a lot to kind of give you a better perspective. So the DNA is your master copy. You do everything in the world to protect your master copy. In the old days, buying a computer program, you would get a CD. Then you would take that CD and load the program onto your hard drive. So if anything ever happened to your hard drive, you could always go back to your CD. That was your master copy. You tried not to use it, you protected it. And that's what the DNA is, that's found in the nucleus. So how do you get the message, the information from the DNA to the rest of the cell? Well, you make messenger RNA. So messenger RNA is made in the nucleus, it comes outside the nucleus, and it gets converted into protein. Okay? So let's look at some of the enzymes here. So when DNA is turned into DNA, that's done by DNA polymerase. Why would you do this? Well, during cell division. Then when DNA gets transcribed into a different language, so the language of RNA, that's done by an enzyme called RNA polymerase. Then when the message comes out into the cytoplasm, the RNA is then translated into a different language, the language of proteins, which is amino acids. And that's done by the ribosomes. Okay, so ribosomes are enzymes, so are those. So the master copy contains both exons and introns. Exons code for amino acids, end up in the protein. Introns do not. So immature messenger RNA is sometimes called heteronuclear RNA, and it contains introns. So these are sections of the DNA that do not code for protein, but they are found within the gene. So the immature messenger RNA has to be processed into mature messenger RNA in the nucleus before it comes out into the cytoplasm. So processing includes removing the introns, adding a poly-A tail, polyadenine tail at the 3' end, and at the 5' end, there's a GTP that's kind of put on backwards, making it so that you can tell which is the 5' and 3' end. And many people believe that this processing helped distinguish cellular messenger RNA from foreign bacteria and viral messenger RNA. So we're going to look at a bunch of stuff, lots of processes, replication, all these different things. And I want you to realize one thing that's kind of important. The knowledge that we're going to be talking about is generalized. It comes from research on all kinds of different species. So the number of proteins involved, the size of things, all this is a generalization based upon specifics coming out of different organisms. So when it comes to questions I'll be asking you on an exam or something, I'm not going to ask you how many proteins are involved or something like that, because those really are species dependent. Anyway, so DNA replication. This is called mitosis, or asexual cell division. So first of all, the requirement is that the cell has to have lots of energy. You saw that the monomers were actually triphosphates, similar in energy to ATP. And so if the cell doesn't have a high energy state, it's not going to have a whole bunch of these monomers. It's not going to go through cell division. And the other thing about cell division is this. If you start the process of cell division and something goes wrong, you're like dead. I was on a PhD committee one time, probably 10 years ago, and that's what they were looking at. They were looking at some of the enzymes involved and what has to happen before the cell division really is a go. And it was the most difficult PhD dissertation I ever served on. It was just so complex. But what I got out of it is that there's so many checkpoints that have to be completed before this actually goes. It's one of the most dangerous things a cell can ever do. So you have the replisome that starts the replication fork and performs replication. And that's a concept pieced together from various sources of research. It's about the size of a ribosome. It's huge. You can actually see it in the light microscope. You need initiator proteins. You need single-strand binding proteins to hold the helix open. Then there's a sliding clamp protein to separate the strands. Then you need a DNA polymerase to ligate the monomers, a DNA gyrase, and this has several different names, to relieve supercoiling. And then you need an RNA primase, and again, this has a couple of different names. And then you need DNA ligase to seal the Okazaki fragments. So let's look at figure 6 if I can find it. So here's the original strand. So notice that way over here, we had to separate those two pieces. We had to break all those hydrogen bonds. And keep in mind, if you were to just take a piece of DNA at the end and pull it apart like that and let go, it's going to wind right back together again. So this whole replisome right here has to work in tandem. So here's what's happening. Here's DNA gyrase that's unwinding the thing. Here's helicase or primase that puts an RNA primer right here. And here's the single strand binding proteins that keep it from falling back together again. And these are the sliding clamps that basically you're allowed to push in that direction. So those are kind of the players. Now let's look at what's happening. DNA polymerase, RNA polymerase, all these enzymes work in the 5' to the 3' direction. So here's the 5' strand. So you started right here and you were polymerizing in this direction. See that? Now look at the other side. This was the 3', right? So now you had to go in this direction. See that arrow right there? So when this is open, it's easy to see that, oh, DNA polymerase starts here and it goes from the 5' to 3' direction. But when this replication fork moves this way, there's going to be a gap right there. So that gap, which is because DNA polymerase has to work stepwise going in that direction, creates what's called Okazaki strands, Okazaki fragments, that then have to be ligated together. So the Okazaki fragments are here, but they're not ligated together. So that's another role of DNA ligase, as opposed to DNA polymerase, is it ligates those strands into one long one again. This is also the reason why we have the terms a leading strand, because here you can simply keep going until you're done. And the lagging strand is done in segments. As this opens, then I can come over here and add more polymerase in the 5' to 3' direction. So going back here, kind of showed you what's happening here in a more simplistic way. So replication is going in that direction. So here's the leading strand. Here the 3' is used as the template. What makes this particular monomer line up right here across from that A? Well, it's attractive forces, which are going to be Londons, they're going to be dipole to dipole, they're going to be hydrogen bonding. In this case, it's hard to say ion to dipole, but I'm sure it probably has some effect. So you get maximum hydrogen bonding between an A and a T, between a T and an A. See how that works? So these things line themselves up according to attractive forces. And then DNA polymerase comes along and says, oh, okay, you guys are aligned, I make the bond. So the leading strand can keep going as the replication fork opens up. But what about the lagging strand? It goes in the 5' to 3' direction. So here's a G, across from that should be a C. So it does the same thing and makes sure all these are aligned and valid before it makes the polymerization. Then eventually, this whole piece of DNA is over here, this whole piece of DNA is over here, and then the cell divides. So notice that this was pre-existing. So they call that a mother strand. This is new, so they call that the daughter strand. So each new cell has a duplex DNA made up of one strand from the mother, one strand from the daughter. And these two cells should be identical. So the energy to synthesize new DNA comes from the hydrolysis of phosphoanhydride bonds by DNA polymerase, which is a ligase. And then also from the hydrolysis of pyrophosphate. So if this were in equilibrium, removing this makes it so that it's almost impossible to go backwards. It's almost impossible to go backwards anyway because you're breaking phosphoanhydride bonds. That's why it's called a ligase. Okay. So in eukaryotes, here's some other pieces of information. Each new piece of DNA duplex is identical. It's called semi-conservative because there's one new strand and one old strand in each cell. And they estimate that it would take about 40 days to duplicate eukaryote DNA with a single replication fork. So eukaryotes have multiple replication forks. They're called replicons. They're bidirectional, and it takes about a day. So when a chromosome is just replicated, the two sister chromosomes are connected at a region called the centromere. That's a picture of it right there. And this is called a chromatid. And the chromatids are separated into individual daughter chromosomes as the cell divides. In asexual cell division, the two chromatids are identical. In sexual reproduction, or meiosis, the paternal chromatid and the maternal chromatid are also joined by a centromere, but they are not identical. And they can actually exchange pieces of DNA between them. And then in eukaryotes, we have what's called telomeres. So telomeres are a region of repetitive DNA sequences at the end of each chromatid. It doesn't code for protein. What it does is protects the chromosome from damage and from fusion with other chromosomes. And then they often are formed in loops, which attract capping proteins that protect the chromosome ends from DNA repair enzymes, all kinds of other things that could actually alter them. Because the purpose of asexual reproduction is to have two identical cells. Okay? So here's the problem, is with DNA polymerase. DNA polymerase can't get to the exact end of the piece of DNA because there's nothing to hold on to. And so because of that, there's an enzyme called telomerase in some types of cells, but telomeres only allow for about 50 to 70 cell divisions before genes start getting lost, and then the cell could end up dying. And this is the case of most of our cells, which are somatic cells. They have a limited lifetime, and then they die. Okay? It's a form of senescence. But some cell types, there's telomerase enzyme. Telomerase is a ribonucleoprotein. So it's an enzyme, has some ribose on it, and it prevents the loss of telomeres during cell division. So what it does is it adds a section of DNA next to the 3' end using its own RNA as a template. It's a reverse transcriptase. So it converts this RNA sequence into a DNA sequence that it then extends the telomere. And then DNA polymerase can go to the end of it. So the telomerase extension is repeated to get sufficient telomeres. Actually, this won't be the right chapter. I changed the chapters because I added another chapter on cancer. So I took chapter 33 and I took out the part about viruses and made another chapter, and I took out the part on cancer and made a new chapter. So I'll have to change this. This is probably 35, I think. Okay. So let's go look at figure 7 here. So here's a rough idea of how telomerase works, okay? So it binds to the 3' end. So here's the 3' end over here. This would be the 5' end, right? So DNA polymerase had gone this way, and then it runs out of space to hold on to. Okay, so telomerase binds over here, and it takes its sequence that it has within itself. That's why it's called a nucleoprotein, ribonucleoprotein. You can see that it's ribose because it doesn't have T in there. It has only A, right? You don't see a T in there. You see, U over there. So across from A is a U instead of across from A is a T. So then it uses this to bring in new DNA. Okay, so across an A will be a T, right? Across a U will be an A because a T is replaced by U in RNA. Okay, and then telomerase moves out of the way, and then DNA polymerase can bind, and then it says, oh, now I know what to do. I start putting DNA there. Okay. Keep hitting the wrong button. So hopefully you get the idea of what telomerase does. So the kind of cells that have telomerase activity are stem cells, cancer cells. Those are the most, for the most part. Okay. So mutation, which would be a mistake in the primary structure, may occur about every billion replications. And that seems extremely efficient until you count the number of cells in your body. So DNA polymerase has another function, and that's checking the DNA for mistakes. So replication goes in the 5' to 3' direction only. So at any time, DNA polymerase cruising around on a piece of DNA, going in the 3' to 5' direction, if it finds any mistake, which means it's a base complementary issue. In other words, across from an A is not a T, it might be a C or a G. It's going to recognize that. And that mismatch will be repaired. But what happens if DNA polymerase, which is not a computer, is simply just an enzyme, what if it doesn't know which one is correct and which one is incorrect? Then that's going to end up making a mutation just because of the repair. So that's called misrepair. Most mutations are detrimental, lead to cell death. But you need a normal mutation rate in order to get evolution to proceed. And this is the reason why we have sexual reproduction. Sexual reproduction is where you get variation in the DNA sequence, so evolution can occur. But you don't want that to have going on during cell division. Now let's look at prokaryotes. Prokaryote reproduction is extremely simple. Binary fission results in 2 identical cells because the DNA is circular. You don't have to worry about telomerase or anything else like that. And because of that, they're not senescent. They're immortal cells. You can keep growing them forever and ever and ever. But still, DNA polymerase in bacteria goes in the 5' to 3' direction, and it keeps going until it's complete. There's no supercoiling, there's no telomeres. Plasmids, which are circular pieces of DNA, are also replicated in the same process. And yeah, so then the cell just divides. Okay, so what about protein synthesis? Protein synthesis is extremely complex in a way, but it has to be perfect. It has to be done really, really rapidly. So what are the major things here to think about? So the gene, right? Well, we know about that. Here's some differences between DNA and RNA. So RNA is 2'-deoxyribose. DNA is 2'-deoxy. RNA is this ribose. Notice that the T in DNA is a U in RNA. This is simplex. This means it's single-stranded. This is duplex. This is only in the nucleus. This is in the nucleus and the cytoplasm. Then we have three major types of RNA, all involved in protein synthesis. So the first one I'm going to look at is messenger RNA. So messenger RNA represents only 5% of cellular RNA at a given time. And anyway, the lifetime of messenger RNA is short because it's degraded by an enzyme called RNase. So remember we were talking about in chapter 31, cell signaling has to be rapid. It's on and then off. And if you want a persistent signal, you have to have a consistent source of that cause, right? And it's the same thing with messenger RNA. It's a signaling molecule. So the gene, the DNA, is signaling the cell to make protein. So the longer this is there, the more protein you'll make. So how does that affect it? What things affect the concentration of messenger RNA? Its production, its rate of production, and its rate of degradation. And RNase is simply everywhere. And then we have transfer RNA, which is about 15% of cellular DNA. So what does it do? It transfers the specified amino acid to the ribosome according to the genetic code. It's the translating device to translate the language of RNA into the language of protein. And so each amino acid has at least one tRNA to bond with it. Some has more than one. Okay. And so here's an interesting case of looking at enzyme specificity. You need at least 20 different enzymes called amino-acyl tRNA synthetases. So these ligases are stereospecific and is sometimes referred to as the second genetic code. So here's the tRNA that codes for methionine, okay? Here's methionine, the amino acid. ATP is required. So what does all this is the amino-acyl tRNA synthetase. So it puts methionine onto only the tRNA made for methionine, and it hydrolyzes into AMP, and then this ends up eventually turning into phosphate anyway. So you greatly ensure that this process is one directional. So this sequence right here codes for this particular amino acid. So this has to bind the amino acid. It has to recognize that specific tRNA, so it has to be able to read that. And what does it do eventually at the ribosome? This is the anticodon for that codon in the messenger RNA. That's how the whole thing works. Okay. Matter of fact, I should probably let you know that this is messenger RNA here. And then the third type is ribosomal RNA. It makes up 80% of the cellular RNA because this is not made to be a message or anything. This is just part of the machine, intimately related to the ribosome. So what it does is it helps the messenger RNA line up correctly on the ribosome. So ribosomal RNA is transcribed and assembled in the nucleolus, or the nucleole, which is a non-membrane bound structure composed of proteins and nucleic acids found in the nucleus. So the product of this gene that codes for ribosomal RNA is RNA. It's not protein. So some people think that, or have been told, that the product of every gene is a protein. Not always. Sometimes it's ribosomal RNA. Similar for tRNA. So yes, sometimes the product of a gene is RNA and not protein. So immature ribosomal RNA has an intron. And that intron is removed by itself. This is the reason why sometimes you will see the word ribozyme. So here is RNA acting like an enzyme. It works on itself. So here's some other types of RNA found in small amounts. Small nuclear RNA combines with proteins to form small nuclear ribonucleoprotein particles, or spliceosomes. And that helps take out the introns from immature messenger RNA. Then we have microRNA, which affects the expression of developmental genes. And then we have small interfering RNA, similar, but blocks expression of a gene. Anyway, so these two are thought to be different, but there are now a few drugs that are based upon this concept right here. And I hope to have that in the chapter on disease. But the major ones are those first three, right, that we need to deal with right now. Oops. So then another factor is the ribosomes. So it's a large complex of protein and ribosomal RNA. They're ligases, form the peptide bond. And they're found in the cytoplasm, where they synthesize water-soluble proteins. They're found on the rough endoplasmic reticulum, where they synthesize lipid-soluble proteins. They all have a small subunit and a large subunit that are apart and non-functional until a messenger RNA starts the process of binding them together. So they're about 2/3 ribosomal RNA and about 1/3 protein. And yeah, they're slightly different between eukaryotes and prokaryotes. So because eukaryotes are always being attacked by prokaryotes, it's logical to assume that the different ribosomes and the different messenger RNA processes are meant for eukaryote cells to protect themselves from bacterial DNA and RNA. So any questions so far? So the first part is transcription. So I separate it into a couple steps. Transcription is making a messenger RNA. It's also done in the 5' to 3' direction. What do you need? You need RNA polymerase. you need transcription factors, and you need helicase. Okay. So RNA polymerases are very large proteins. There's a couple different kinds of them, which you don't need to be worried about. But transcription is way less complicated than replication. So I drew this kind of thing right here. It's not, not publication quality, of course. But think about DNA replication. Essentially, you have this twine made up of two strands, and you have to completely separate those two strands. It's like taking a piece of string and pulling it apart like this. What you're going to get is all this unraveling. It's going to get kinked up, it's going to get messed up, and you have to separate all that stuff that's going to get all knotted. When it comes to transcription, you do not have to separate the pieces of DNA. It's like you take a pencil and you stick it in there, and then you just wind the pencil around wherever part you want to get to. And as you move from one part towards the other part simply reseals itself behind it. The other thing is this. What if you make a mistake in the protein? Is that really that deadly of a mistake? No, because that protein is not going to last forever. If you make a mistake in DNA replication, that mistake is going to last forever, so it could easily kill that cell or cause something like cancer. So again, RNA polymerase goes in the 5' to 3' direction. It does not check for mistakes. And you may get an error about every, 10,000 transcriptions, but no big deal. The other thing, that there are some extremely potent toxins that inhibit the RNA polymerases. One thing that maybe you should think about: if for some reason an organism was not able to synthesize new proteins, that organism is going to be dead within a matter of minutes. So anyway, here's the coding strand. The coding strand is the gene. And then here's the template strand over here. So that's part of the DNA. Notice I put ATG, and across from it would be TAC, right? So RNA polymerase goes in the 5' to 3' direction. What's it going to code from? This AUG is the same information that's in the coding strand, right? Except U replaces T in RNA. So the messenger RNA represents the same information that's in the coding strand, which is the actual gene. But how do we make this complementary information? Is using the template strand. So across from a T would be an A, across from an A would be a U, across from a C would be a G. So here's the DNA coding strand, here's the template strand, here's the actual messenger RNA. You see how the messenger RNA was made complementary to the template strand so that it would contain the same information as the coding strand. And then, how does this get translated into protein? By the ribosome. Well, AUG codes for the amino acid methionine. UGG codes for the amino acid tryptophan. CCC codes for the amino acid proline. And notice that the convention is, when we write DNA there's a single strand, the 5' is always on the left. Go back to when we studied proteins. The amino terminus was always on the left. So here's the reasons for those conventions. So then messenger RNA processing is called post-transcriptional modifications. This occurs in the nucleus of eukaryotes. So how does this happen? So the spliceosome is composed of small nuclear ribonucleoproteins called SNRPs, some other proteins. So here's the message. They took out the introns already. Here's the stop codon, and after the stop codon is this poly A tail that was put on. And then here's AUG was the start codon. And then a GTP was put on backwards. It's methylated, so it's not exactly GTP. And this is what works with eukaryotic ribosomes. And if you had a prokaryote messenger RNA, you might have AUG. Matter of fact, you would, but you wouldn't have that cap, and you wouldn't have the trailer on there. So speaking of introns, here's something really kind of interesting. A single gene can be expressed as several different proteins depending upon how the introns are spliced out. It's called alternative splicing. So we know that there's a single gene in the rat that expresses 7 tissue-specific variations of the muscle protein, tropomyosin. So an exon in one cell type can be an intron in another. Does that make sense to you? So the number of genes in an organism's genome does not by itself tell everything about the complexity of the organism. And it also brings up another issue. If introns are removed by the spliceosome, who tells the spliceosome what to do? This would be gene regulation, wouldn't it? So gene regulation is much more complicated than we actually think. So the nucleus is encased in a double membrane called the nuclear envelope. The outer membrane is fused with the endoplasmic reticulum, and only certain molecules can go in and out of the nucleus through the nuclear pores. So for example, ribosomal RNA is produced in the nucleolus, and ribosomal proteins brought in from the cytoplasm bind to the ribosomal RNA, and then the ribonucleoprotein complex can enter the cytoplasm. It's a rather complex system that's all designed to limit the number of proteins and anything else that's allowed to interact with your DNA. Okay, let's see. Is there something else I should have been looking at here? Guess not. Okay, so let's look at figure 11. Oh yeah, here's figure 9. So here it shows you the messenger RNA Here's RNA polymerase going in the 5' to 3' direction. Okay. Termination site, that's the stop codon. Stop codon in the DNA. So here is a gene and a bunch of introns in there. So as it's being processed, here's the heteronuclear RNA, which is immature messenger RNA. The poly A tail is put on there. You cut, hey, what happened? You cut out the introns, right? And you ligate them together, and then you add this GTP cap, and then it can come out into the cytoplasm. What I was talking about with the rat gene for tropomyosin, one type of tissue would take out this one, and another one might take out this one and that one, giving you different types of tropomyosin. So this is actually more complex than I want you to know. But right now, you can see that the small subunit and the large subunit, whose large subunit is apart. So here's a sequence in the beginning of the messenger RNA, and look what has to happen. Initiation factor 1, 3, don't worry about that stuff. Okay, all you need to know is that there are such things as initiation factors. You need to know that it requires GTP, and then you need to know that you need the first loaded up tRNA. So when you have all these things together at binding, then the large subunit can then bind to the small subunit. Okay. So what do you need? The messenger RNA, you need some initiation factors, you need GTP, you need the first loaded up messenger RNA that basically codes for start codon. And then some factors come off, GTP is hydrolyzed, and now you're ready to go. Oops. This is the initiation complex. Everything is all together now. And then what happens? Okay, here's the growing peptide right here. So if you look at all this loop and everything over here, you have elongation factors, you have GTP required everywhere. But essentially what's happening is that the new loaded up tRNA comes in and binds to the site over here on the messenger RNA. So notice if this is C, I can't read that directly, G, C, yeah, here's the complementary sequence right here. So how does the code in the messenger RNA give the sequence of amino acids? It's all by the hydrogen bonding that's possible between each codon and each of the anticodons on the tRNA. So notice everything is lined up now. And then what happens is kind of what you don't expect to happen. Notice that this was what was here first, right? What was here first, what was existing, is then transferred to what just came in. Okay. See that? So we started off with methionine. That was the beginning of the polypeptide, right? And then the next amino acid came in, and the polypeptide that was there on the ribosome got transferred to the new incoming tRNA. So notice you need elongation factors, right? GTP is hydrolyzed. Okay. So that keeps going until you reach the stop codon. So this is the way an organic chemist would look at it, okay? So here's the original one, right? And what's happening here is that this amino group is transferred to the next one. And what they don't show you is that this is isoleucine. They don't show you how this one came in over there, but that's okay. But here's the hydrogen bonding. Okay. Okay, so let's go through this. So the activation step. We talked about that in the initiation step. What do I want you to know? Well, I want you to know some of the things that are required for initiation. With elongation step, what do I want you to know? Some of the things that are involved for elongation, and that there is an elongation complex. So then finally, what do you need to know about termination? When you reach the stop codon, of which there is at least 3, you need release factors, you need GTP, and the complex is finally cleaved from the last tRNA. There's a peptidyl site. I don't care if you know about that. There's the acyl binding site. I don't care if you know about that. And again, the reason is because what we know is simply a conglomerate coming from all different species. Okay, so any questions so far? I know it's complex, but each one of you is doing this right now as we talk. I might even be doing it at a much slower rate. So here's a couple other things. So polysomes. What the heck is a polysome? Well, originally they noted that these were like acting like beads on a string. In the light microscope, you can actually see ribosomes. You can't see the messenger RNA or the proteins that they make. But they found these beads and they were like bouncing all around due to Brownian motion in the microscope. They all seem to be connected to each other. So what it was is that you have several ribosomes are able to line up on a single piece of messenger RNA and start making protein because they all go in the 5' to 3' direction. So what that does is it greatly amplifies the number of proteins that you can get from a single piece of messenger RNA. And what we learned from the chapter on proteins is that secondary structure is determined by primary structure. Secondary structure determines tertiary structure. So you can see this one right here is already forming secondary and tertiary structure, and it's not even finished yet. Everything is driven by what? Intermolecular attractive forces. And then we have post-translational modifications. So as this process is going on, or even as it's over, chaperones help fold the protein correctly. In mammals, the first methionine, which is the start codon, is usually hydrolyzed off. It's gone. Disulfide bonds are formed by oxidoreductase enzymes. Prosthetic groups are added by transferase enzymes. Amino acids can be modified by transferases, add functional groups, and then quaternary structure forms. So notice that we had post-transcriptional modifications and we have post-translational modifications. And both of those make it very difficult to take a protein and go back and get the sequence of DNA, or to take messenger RNA and go back and get the exact sequence in the DNA because of those processes. So cellular proteins are synthesized by free ribosomes, usually attached to the cytoskeleton somewhere. And proteins made for export are synthesized by ribosomes on the membranes of the rough endoplasmic reticulum. They pass through the Golgi apparatus, get processed, and occasionally accumulation of improperly processed proteins in the ER is associated with like Parkinson's and in Alzheimer's diseases. Figure 14. Okay. It's our last figure right here. Okay, so here's the nucleus. Here's the rough endoplasmic reticulum over here. And so membrane-bound proteins, which would be not very soluble in water, but meant to be in the membrane, are synthesized in the membrane, and they're transported by vesicles into where are we over here? They're transported in vesicles to be processed, and they end up by exocytosis of secretory products. If the membrane, this vesicle basically fuses with the membrane of the cell and empties its contents, or these proteins may be meant to be on this membrane surface right there. So when they fuse with it, then there they are. So toxins and antibiotics. So I went through several of these. I don't want you to memorize all this stuff, but let's think about this. The diphtheria toxin inhibits the peptide from being transferred from the peptidyl to the acyl site on the site of elongation on the ribosome. Okay. Ricin, one of the most dangerous naturally occurring toxins. So okay, where did I put the? Where did I put that? Oh, up here. Okay. Block formation formation of the initiation complex. So how long does it take for you to die from ricin or any of these things? Not very long at all. Okay. So here's some that work on bacterial ribosomes, but they don't work on eukaryote ribosomes. That's why we can use them as drugs, right? Okay. So one of the most complicated things that the humans have ever tried to figure out is gene regulations in higher organisms. Gene regulation is extremely difficult. So first we'll look at what we know about prokaryotes because it's rather simple. So it's called an operon. So here's an operon over here. So you have a regulatory gene that codes for the operon's repressor protein that makes it turn off. The operator gene is where the repressor proteins binds to turn it off. The promoter site is where RNA polymerase would like to bind. And the structural genes are simply what would be turned into messenger RNA. So the best known example is the lac operon. So if you're a bacteria, you can't go to the food store and say, oh, I want some lactose to eat today. You don't get that choice. but you have an enzyme that allows you to break down lactose for energy. So most bacteria may never come in contact with lactose, but if they do, they want to be able to take advantage of it as a food source. So most of the time, that gene is turned off. So here's the situation. Here's the regulatory gene that codes for the repressor protein. Here's the operator site for where the repressor protein binds. And here's RNA polymerase that wants to bind here, but it can't get there because of the repressor protein. So this is off. Then how does it get turned on? An inducer, in this case lactose, binds to the repressor protein and causes it to change its conformational shape. That then causes the repressor protein to drop off. RNA polymerase can now bind to its site, which is the promoter site, and then it simply proceeds in the 5' to 3' direction, making the messenger RNA for lactase. So what happens when you've used up all the lactose? Here's an equilibrium right here, a simple equilibrium. If lactose disappears, right? If you remove this, what's going to happen is this is going to start turning into the native form, that conformation. And what can it do? Bind to the operator site, making an RNA polymerase unable to bind to it. Now the gene is off again. So that's simple. What about in eukaryotes? Well, we have repression of genes, we have induction of genes, but we have no clear example of an operon. It's much more complicated. And so all I'm going to try to do is show you why it's so much more complicated than in a prokaryote. One is that genes are not grouped into operons. So hemoglobin has alpha and beta subunits, and the genes for them are found completely on different chromosomes. Like, what the heck? That means that every time you make an alpha, you need to make a beta. But they're on completely different chromosomes. How are you going to organize that? Then most genes in adults are turned off. We have developmental genes that are only used in certain stages of development, never to be used again. So here's a kind of a cute example of otters. Those are developmental genes. And then we have differentiation of cells. So skin cell has the same gene for peptidase as basically your stomach cells do, but it never expresses. Otherwise, a newborn would be digested by its mother. Hormones and other effectors regulate genes. Okay. Viruses and other traumatic events can turn on or turn off genes. Then telomeres are found only in eukaryotes. Prokaryote DNA is circular. And then in certain white blood cells, the antigen presenting cells, they're licensed to do genetic engineering. They're producing brand new proteins that have never been in your body before. And then we have regulation at the level of transcription by general transcription factors. We know of at least 6. So enhancer DNA sequences can be used to speed up transcription. Silencer DNA sequence can be used to inhibit transcription. Response elements. And you know what? Many of these are not in what's tested for in gene testing. Okay. The simple gene testing is not going to do everything we need it to do. Also in eukaryotes, genes are protected by histone. And so you hear the word remodeling of DNA, of chromosomes, because it's just simply complex. How do you get to the gene when the gene is just simply so buried and protected? So we have certain cells that, certain proteins that are known about that that produce these non-coding RNAs. That means that they don't produce proteins, right? Yeah, so remodeling complexes, that's something that, yeah, should be known. Then there's cohesion loops. So under the microscope, the cell's DNA looks like it's just total chaos. And these things can enhance each other. So here's a way to bring genes closer to each other that are spread out on the chromosome. So it's just crazy. And then we have long non-coding RNA. So look at this. Here's the gene expression over here. Here's the polymerase complex. Here's a chromatin modifier. In other words, it's modifying the shape of the chromosome in that area. Here's a transcription factor, and here's long non-coding RNA that seems to be like, what the heck? It holds this complex together. If you don't have it, then you can't open up, the gene and get RNA polymerase into it. Some genes are regulated by post-transcriptional level between transcription and translation, maybe by microRNA. And they found this one particular group of small GTPases. And the reason why this is important is because when these are overactive, that correlates to cancer, several types of cancer. And so, anyway, there's the KRAS gene that codes for these small GTPases. It's turned off by this microRNA they call it LET7. And there's several drugs that are now being tested to attack these genes and turn them off. Okay. So here's normal protein production, right? You get normal cell growth. So here's your small RNA phosphatases or kinases, ATPases. And if this is normal over here, but if you have a variant gene over here, this LET7 can't bind there. So these genes are just overactive, producing too much of these proteins, leading to malignancy over there. So this is kind of interesting that the co-discoverers of this relationship right here, they didn't want to give it to the big drug companies. So they wanted to just make it as a service to mankind. So they formed a nonprofit. Anyway, that's pretty interesting. And humans just simply have a lot of DNA. So then we have the Human Genome Project, and we find that mutations associated with diseases are actually outside, oftentimes, of what the actual genes are, making things much more complicated. And then we have epigenetics. So epigenetics means beyond genetics. So it's not simply modifying the primary structure of the DNA. And so, for one, about half of the human genes have promoter sites that are rich in GC. They call these cytosine, phosphate, guanine islands, right? I don't know why I started. They used to just call them GC-rich promoter sites. Now they call them these islands, right? And a lot of these get methylated or are not methylated. So what does that mean? You take the C and you methylate it, you turn off that gene. Okay, so now you have gene regulation by a methyltransferase. But who regulates that methyltransferase now? It just gets more and more complicated. So when we come back tomorrow, which will be again at noon, we can start with epigenetics, and that's kind of interesting. I think we're kind of out of time right now. Anybody have any questions at all? So if you haven't taken your exam yet, you need to do that today. So while I was lecturing, I was watching for any incoming emails in case somebody wanted to start the exam, but I didn't get any. So any other questions you might have?
No questions right now.
Okay. Well, you can always email. And,
We can email you around 5 o'clock, and that's fine for the test too.
Sure. Yep.
Okay, thank you.
Okay, well, you guys have a great day, and I'll get this lecture up as soon as I can. And take care.
Thanks, you too.
Thank you.